Hello, I'm Dr. Frederic Tolozan. Welcome to our Reinnervate product training sessions. So today we are going to concentrate on Alvetex three-dimensional cell culture technology. I work in research and development for Reinnervate, and I also am the Alvetex support specialist. And just to give you a little bit of background about myself, I have a background in cell biology, and I am especially interested in interaction between cells and their environment and how that affects their behavior, which is very important in terms of function, by example in terms of drug resistance. And this is how I was naturally attracted to work with three-dimensional culture and Alvetex. So to get started, let's have a look at that. Come on you. There we go. So we will go through different sections. First, I will go a little bit into explaining what is 2D culture and why 3D is better than 2D, D being for dimensional. A bit more details about what we mean by three-dimensional cell culture, and why the company has developed Alvetex in particular as a solution for routine 3D cell culture. And this is very important because we wanted a product that would be convenient and consistent so that academics and industry people could trust and could use routinely in the lab. Alvetex has specific designs, functionality, and a lot of applications, very wide number of applications. So we'll just give you an example. A few examples of key applications, things that are more popular with our customers, and to give you an idea of the range of work that you can do with Alvetex and how useful it is. So to get started, this is in vivo tissue, and you can see that in this natural real tissue environment. Here are the little cell nuclei, and you can see how all the cells are really in contact with each other. And in between each cell you will find some extracellular matrix, so that is protein which is secreted by the cells and gives a cohesive cohesion to the tissue and to the organ. And here it is detailed a little bit more. Now, when we take those cells and we study them in the lab, we dissociate them from that extracellular matrix and protein coating which holds them together, and we end up with individual cells which we then are going to grow in the lab outside the body. This is what we mean by in vitro, which means in glass. And really, most of the time it's actually in plastic rather than glass. So in two-dimensional cell culture, and here you will see some format that I'm sure most of you would be very familiar with. We take those cells which tends to be quite cuboidal, quite homogeneous in their three-dimensional shape, and you put them on a surface, which is a flat surface here, which tends to be plastic. Now, when the cell goes onto that substrate, it's going to detect that it's got a flat, rigid surface at just one end, and it will adapt by expressing proteins, most likely cytoskeletal proteins and anchorage protein, which are going to help adhere to that substrate. And it is going to do that more and more through feedback system to the nuclei, to the nucleus, so you have more gene expression, more protein production, and you get more and more adhesion of the cell, up to the point where you end up with a very flat cell, which is quite different from the morphology it would have in vivo. Here you can see this is a bright field image, and you can see a lot of little nuclei. So from the top it looks quite similar to what we had in vivo, but if you were to look on a side view you would see that the cell is actually very flattened. And if we go to the next slide, so just to recapitulate, here is the cells in vivo, so it's very spheroid really, and it's got a lot of contact. And when you compare it to 2D, you can imagine how this is really just the only surface along which this cell is going to have a contact with its neighbor. Most of the surface is in contact either with a substrate or with the medium. And you can imagine how that is a very, very different environment compared to what the cells are used to here in the real tissue. Now, what we have achieved with Alvetex is that we have managed to reconstitute an environment where the cells here grown in Alvetex are a lot more in contact with the neighbors, and this is therefore a lot more like a three-dimensional in vivo environment. And when you get the morphology looking like in vivo, you're much more likely to have the function of the cells also being like in vivo, which is more relevant for what we want to do as scientists. So how do we define three-dimensional cell culture? Well, a lot of what I've explained is about maintaining that normal three-dimensional shape that we can see, and the shape is obviously supported by intracellular structures and organization of proteins. By keeping that three-dimensional shape, we encourage the cells to have more cell-cell contact with the neighbors. That means it allows them to form complex interaction with adjacent cells, which means more signaling. And if you have more signaling, again, you can have cell behavior which is more like what you would see in vivo. And the whole result is you're getting a more natural environment. And this is what we want. We want restitution of native architecture, so the tissue that you're creating in reconstituting rather than creating in vitro is more like in vivo. And we reduce essentially a lot of artificial responses that are associated with 2D. When the cells are flattened, as they are in 2D, that means that their protein intracellular localization has changed, and that is going to affect the cell processes which are happening within the cells, and this is going to have an effect on what you see happening in the experiments. So we are going back from that artificial responses, and by providing them a better environment, hopefully get a more physiological response, which is better translatable compared to in vivo models. So here we're introducing this website, 3dcellculture.com. This is a completely independent website, and they are dedicated to three-dimensional cell culture. And you see what difference it makes. So this is not a commercial bias. Essentially, it's a completely independent body, and they have a lot of information that you can refer to show independent advice as to how 3D cell culture is very much beneficial to cell growth and cell behavior, and therefore to scientific relevance in general. So the three main things we have to think about when we're culturing cells in a three-dimensional environment is cell shape, as I mentioned before. Typical cells in 3Ds are very homogeneous, and they tend to be cuboidal or ellipsoid or spheroid, again 10 to 30. Cells in 2D, much flatter, as I said, 3 microns, so much difference in the shape of the cells, and therefore the internal organization of that cell. As we mentioned before, typical cells in 3D, all of the surface mostly is exposed either to other cells or the matrix, meaning huge amount of signaling either between cells or between matrix and the cells. And by comparison, the cells in 2D, about 50%, slightly less than 50% against the medium, therefore no cell contact, about 50% exposed to the flat culture surface, which means a lot of anchorage and anchorage-dependent mechanism happening, but between a chemical surface rather than a cell to a cell, and only a very, very small percentage exposed to other cells, so a very small area for true cell-cell signaling to be occurring. And as a consequence, the cell behavior in 3D compared to these shows differences in the number of cell behaviors. And all of them, when you have a look at them, you can see at the relevance of these differentiation if you work in stem cell field, drug metabolism, this would be very important for industry, general cell function, in vivo relevance, this is also very important to industry because obviously you want a good translation into your animal models. And so a lot of processes that can be affected and therefore improved when you go from 2D to 3D. And if you have customers in the field that are going to ask you for, oh yeah, you're telling me this, that's very well, but can you give me exact proof? You go to that website which I mentioned earlier, 3D Cell Culture, and they actually keep a repository of papers showing exactly those differences. And it's not specific to our product, it's just 3D Cell Culture in general. And here are just a few examples. Obviously, I'm not going to go through these in details, but it's just to give you an idea of this is a good source if you want just hard evidence and say, I'm not just telling you this, look, scientists have found it in a different system with a range of technologies. So it's a good introduction for customers which are maybe not too familiar with three-dimensional culture. Now 3D cell culture is relatively new in the field, but it is increasing in interest. A lot of it fueled by ethical concerns and wanting to reduce animal use. So anything that is going to increase the relevance of your in vitro use and the translation, yeah, we'll do that, and the translation of your data into in vivo models is very welcome. And as you can see here, this is from cellculture.com again. This is a number of papers mentioning 3D culture methods in general, and you can see how there's been an exponential trend going up, and picking up from Sigmund from 2008, and again you can see there is an increase in the number of papers. That means there is a lot of interest in the field. There might be less people who have actually tried it, but there will certainly be a lot of people that might be wanting to try it and might be unfamiliar with what culture to use, which one to go best. And obviously at the back of that there's a lot of events, more and more events being organized, and a lot of alternative technologies, because it is still quite a very young field. So here are a few examples of well-known companies, a lot of different technology, different type of technologies, and each of them really has got potential limitation compared to ours, obviously. Biodegradable polymers, that can be quite popular with customers who want to do in vivo transplantation, and that is quite a specific application. Now, Alvetex is not biodegradable and it's not approved for use in vivo. But we think that for use in vitro, biodegradable polymers are not ideal because by their very nature they will be unstable and they will degrade over time. Therefore you have questions about shelf life of your product, and also as it degrades, well, what is it releasing in the medium, and is that going to have an effect in your experiment? You don't have that problem with Alvetex because it's extremely stable. In terms of electrospun fibers, fibers are creating a fiber mat. It's a technique which is actually quite difficult to get a consistent mat. We have experience of it, and it is quite a very tricky technique to get right, so you get problem in consistency. The other problem is you do get a mat, i.e. you do get a 2D surface which will have some three-dimensional patterning at the top, so you get micro 3D or micro patterning, but it's not really a surface in which the cells can really invade into, as opposed to Alvetex, which is a void-based structure where the cells can really get into it. The fiber scaffold has got, in a way, the opposite problem compared to the electrospun fiber, which is that the fiber is actually quite large. So you have cells which grow on a curved 2D surface, and it's really only in the places where the fibers meet each other, where you've got a bit of a corner, that you can have a small amount of three-dimensional culture. But by and large, it's mostly a curved 2D culture as opposed to a 3D culture. So again, not genuine 3D. And then you've got scaffold made out of different type of products, alginate, so this is a plant-based product. And again, you have issue with stability of the product, and when you've got specific, well, this is a chemical that might have effects with your cells, so it's not always the best one, depending on what cells you're going to grow into, and you've got problems of stability as well. But this is an alternative which can be quite popular, aside from collagen, which is already best known. So we find that most of the problems with our competitors' technology that we can poke at is that, are they getting genuine, real three-dimensional cell culture? And a lot of them don't. It's called 3D because it's an easy way to call it, but when you look at it, it's not actually really three-dimensional. It's generally more like a 2D on a curved surface, on a 2D on an elevated surface, but not really three-dimensional. A lot of them also are a little bit tricky to manipulate or to use or to get consistently, and again, this is something that we really wanted with Alvetex, to do something that anybody could use and was consistent, and it was just nice and easy, and you could, and again, developed for routine use. And because it's quite an early still a young market, as I was showing in the slide before, there is not yet a really well-established historical leader in that field. So customers are presented with really a wealth of options, and it's very much about us to educate them about, well, this is actually maybe not what you want because of this and because of that, and Alvetex can do this and can do that. So I guess a lot of customers will come not having too many preconceptions about this is definitely what they want, and it's up to us to really educate them about all the different little technologies and why ours is a good choice for most options. So next one, there you go. So the next couple of slides, again, I will just go very quickly through these. You can go back through these on your own time. Just list a few examples of products by different companies, and again we've detailed here the potential limitation for routine 3D cell culture compared to our products. So if you encounter a customer in the field which maybe has heard about one of those products or heard of similar technologies, these are the arguments you're going at with them to say, well, actually this is, maybe you want to try ours, is actually going to be better for you. And there's a couple of pages of this. Again, I'm not going to go through these. You can go through these in your own time when you review the publication later. And if you want a visual little video, which you can use for yourself, but also you can direct your customers to, we do have one on our learning zone, which is dedicated to looking at different technologies for 3D cell culture and again the limitations. There's about 4 or 5 in them, the better known ones, and it's a nice little tool, a little visual tool in about 5 or 10 minutes for your customer to really get a grip of what are the main characteristics of each technology and why ours is better, obviously. There you go. So the design features, which is what we really wanted when Alvetex was first introduced, is we needed something that was reproducible and consistent. This is important obviously in industry, but also in academia, because you don't want variability in your product. There's already enough variability in biological system. You don't want to have to worry about your plasticware. So this is something that we can say every time you get an Alvetex, it's going to be exactly the same thing. You don't have to worry about it. It's inert, which means that it's got a very long shelf life. You can have it on your bench for so many months, so many years. You can go back to it. You know it's still going to work. It does not degrade, and it remains stable. It's actually made of exactly the same material as the two-dimensional in vitro, most in vitro products. It's made out of polystyrene, so you know you're using chemically exactly the same thing, and it's a chemical that is going to react with very few other chemicals out there, so it's not going to leach out anything into your culture that you're going to have to worry about. It's adaptable to existing cell culture format. We will go into that in the next slide. And this is something because, again, we wanted scientists which are familiar with using a specific system to be able to just take Alvetex and use it in exactly the same format. Again, reducing the variability, so that's something you don't have to worry about. It's a consumable product. Again, most two-dimensional in vitro product, you would use it once, grow your cells, take your cells back, put it in the bin. Alvetex, it's exactly the same thing. You use it once for your culture, next culture you just order more, which is good for us. And we have a lot of applications. Because we've been in the field now for a number of years, our research and development department through quite a few people which have done an awful lot of work to develop quite a lot of applications now, and we do have a wealth of information and in-depth information to show everything you can do with Alvetex and how you can do it. Because again, it is a new product and people need quite a lot of support to guide them through how you can make the best out of it. And we have a lot of protocols to help them do that. And, whoops, sorry. Can we go back? Yes, we can go back. And the main idea is that you can get data from it that you can easily compare to your 2D, and it's very good for generating a lot of knowledge for your model. So I've been talking about Alvetex. I haven't said yet what it is. Well, you can see here this is an SEM, so scanning electron microscopy and high magnification. That shows you the structure of Alvetex, and it is a HYPE, which is a high internal phase emulsion, And what you have to imagine is, if you imagine quite a lot of bubbles which are stuck together, and then the wall of the bubble is going to solidify. And as it solidifies, it gives the arches that you can see here. So your bubble would be the big things in the middle. And in the spaces where the bubbles were touching each other, we're getting little interconnects, is what we call them. So the cells are going to grow into those big voids, and then they can go from one void to the other using the interconnect. And because we've got the technique in a consistent way, we are able to control the size of those bubbles, which we call voids, and the size of the pores. We're getting a very high porosity, between 90 and 93%, so it's mostly air, i.e. it's mostly surface volume for the cells to be growing in. And in terms of mechanical properties, it's not, you can't squish it. We don't really measure the mechanical property ourselves, although we have at customers, we have measured Young's modulus for it. So if you get any customers which are interested in the mechanical property, we have a little bit of information published on it. But in general, it's not a material that is going to stretch or that is going to deform, which can be quite useful, because if you compare it, by example, to a collagen gel where the cells are going to contract and contract and contract and everything is going to move, Alvetex is not going to move. It's going to be absolutely stable mechanically as well as chemically. So that is another advantage of using Alvetex. Right, so this is a lower magnification, so you're getting a better idea now. We've got that structures that I was talking about earlier, and we manufacture them as discs. So you can see here how the discs are being cut. That is the side to about 100 micron thickness, which is consistent all over the disc and every disc. And each disc is then placed, by example here, at the bottom of a plate. And this is what you get in the product. So you've got your recognizable, well familiar 12-volt plate format here, and at the bottom you are getting your three-dimensional surface, or your three-dimensional volume rather, and this is what the cells are going to grow into. We have this available in different formats, again, because we have to answer quite a lot of different needs from our customers, depending on what they are being used to. In terms of plates, we have 12-volt plates, 24 plates, and really for industry, we've developed the 96-volt plate and the 3/8-volt plate. In all of these formats, the Alvetex is at the bottom. So you're going to have cells growing inside here, and they are being fed from the top. That means you tend to have growth mostly in the top portion of your disc. Now, because we wanted the customers to be able to get the maximum out of 3D culture, we've also developed insert formats. Here you can see you've got a little insert with little wings on the side, and the wings are going to help the insert to sit inside the well, and the insert is being suspended. So you're getting medium coming from the top, which is on the top of the insert here. Can I get to you? Yes, you might be able here to see that you've got little windows which have been cut in the wall of the insert. This is to allow the medium to come in, to float in, so the cells are well fed from the top, but also because the insert are suspended in the well, they are being fed from underneath. So they're getting a maximum amount of nutrition, and this is going to help in terms of growth. Because we have the windows, we also have the option to have 3 different feeding regimes, one which is going to come just at the bottom of the insert. This is of interest for people who want air-liquid interface, like people working in skin, or people who might be working in air exposure, or people working in lung models. Then you can have your medium inside the insert, but just underneath the window. So this is more where you would want to have one medium on one side, one medium on the other. And then you can just go medium over the window, and that's just for maximum growth. So these inserts are available in 6 wells, but also we've got the smaller model. Because this has got larger wings, it can fit in a 6 well, as shown here, but the wings can also be snapped in 2. And in that case, it will fit in a 12 well. So it really depends on what the customer wants. For cells which have really high demands in medium, we've developed this system here, which is a well insert holder. And you can see we've got an extra deep Petri dish with a holder, and then you just put your inserts in there and you can get a lot more amount of medium. So this is where you've got long-term cell culture or cell culture which you don't want to disturb, so you can just put the medium in there and then just leave them for much longer. And then the last one we have, which is an expansion of this really, is a perfusion plate. So here you can see it's a modified 6-well plate. You can have your little insert, and we have windows in between the wells. So we'll start at one end, and medium can be pumped into the first well and just through the whole system and out again. This can be very useful if you have even more medium requirement, because you can have an external medium bottle which is a higher volume with everything just circulating. It can also be useful for model of infection, where if you imagine you had a culture that was infected here, and you could just record how quickly the infection is going to travel through the system. So that's just an example of applications. And all of these we have detailed in the catalogue, and here is the link. And the catalogue is very good because it has very detailed graphics, and they're very clear. So if your customer is not quite sure about, oh, I don't really understand what you mean by the plate, the insert, how does it fit, how do they sell that, you just direct them to the catalogue and they can find really clear graphics of everything, how it fits, and also the product number. So it's useful for them, and it's also useful for you if you want to familiarize yourself a little bit with the different option that we have. Right. In terms of substrate, as I said earlier, we know how to control the size of the voids of the scaffold, so we can have it consistent throughout the scaffold. What we can also do is have it in different pore size. This is Alvetex scaffold, which was the first product that we introduced to the market. And as you can see, the voids here are quite large. They're about 40 microns in diameter. So this is plenty enough to allow cell growth into it. Interconnects about 10-13 microns diameter. Again, more cells will be able to penetrate through those interconnects to invade through the whole of the scaffold. We've recently introduced another product, which is Alvetex Strata, and you can see these are the same magnification, and immediately you can see how the void size is a lot smaller. On average, it's about 5 micron. The interconnects, which are still present in there, are about 5 micron diameters. Obviously, when you put cells on here and there, it's still 3D substrate, but it's going to have a very different behavior because of the difference in void size. We find that more cells will invade inside the scaffold, but with Strata, you can differentiate between cells which will stay on top and which multi-layer, so you still get a three-dimensional culture, but it tends to be substrate-free on top of your strata, and cells which are more invasive, and they will actually manage to get through, but not all the cells will grow inside. So depending on the application of your customers, you might want to direct more toward one product or the other, or even if you're not sure, offering a kit. By offering, I mean offering the option of trying a kit which has got a mix of both, so they can see for themselves which one is working better for the system. Right, so this is a little bit of specification difference between scaffold and strata. Thickness is the same. The disc is cut to exactly the same thickness. As I mentioned before, the difference is in the void sizes and the interconnect sizes, strata being a lot smaller. And the good thing is it's only got a very small effect on the porosity. It's still 90% porous compared to 93% in scaffold. This is a very small difference. And still mostly air and still mostly potential growth volume. I was mentioning, in terms of colonization of the substrate, scaffold, they will go in, and most cell types should go in all the way if you give them enough food. While strata, you can really differentiate between epithelial cell types, which will stay on top, versus invasive cell type, which will go in. One of the good applications for this is for invasion assays. If you want a system which has got a little bit more resolution, which is going to be a little bit more trying on your cells, to differentiate between slightly invasive types and really invasive type, strata is a good idea. In terms of format, the difference with Strata is at the moment we are only offering it in insert formats. So if people are really interested in plates and nothing else, then it's got to be scaffold. Strata is not an option. Not because of the nature of the substrate, but it's because we don't have it available in that format at present. Here again, something you can refer to again later. I'm not going to go through in detail through these, but these are the type of assays that can be done in scaffold and strata, and that gives you just an idea of how versatile the product is and how wide and routine the applications are. And yes, we can do it. A lot of people will ask you, can you do protein expression? Can you do RNA? Can you do this? Can you do that? The answer to most of their question is going to be yes, you can. You might need to check our protocol to make sure you're doing it right, because we'll have a little tricks to make sure it works. But most things, yes, you can do both in scaffold and strata. As you can see here, again, Alvetex strata is only available in inserts. Compared to scaffold, which has got plates. And then we have applications which are suited better to one system than the other. Obviously, air-liquid interface is only going to happen in insert. You're not going to be able to do it in plate. Permeability, for the same reason, only going to be able in inserts. Strata is better for things like invasion assays. Again, depending on your cell type, but if you want to differentiate between slightly invasive and very invasive, strata is a good idea. If you want to do ex vivo tissue maintenance, this is where you are explanting a piece of tissue and you want to leave it to observe it or to test it. But what you're really after is viability. If you just want the tissue to be viable and to be stable, then strata is the best thing, because it's just going to sit on top and adhere to it quite well, as I will show you later, because it still has got that 3D patterning surface that the cells can really grab onto, but it's just going to stay there. If by comparison you want to take ex vivo tissue and you want cells then to kind of move away from it and invade into a substrate, then scaffold is a good option. So it's talking about with the customer about their application and exactly what they want from their assays, and that should help to guide you in recommending is a strata or scaffold. Ooh, come on. Next slide. Ooh, maybe that's a big slide. It's just waiting for it. There we go. Right. In terms of cells, which we know grow in Alvetex, because that is a question we get quite often from customers who go, well, I've got such type of cell, is it going to work? We have grown personally in R&D, myself and a lot of other people, quite a lot of cell lines, and a lot of them take through it. We've had one cell line that didn't like it. And then we've had customers which have just gone and done their own things, and again, this is already a long list of cells which, yes, you can grow in it, but it's a non-exhaustive list. We have more. So by and large, cells are quite happy to grow in Alvetex. They will just take to it, and whatever the customer is wanting to grow, chances are it will. And only thing that to look out for is some blood cells, which can be very, very small. So if the cells are very small, you might want to recommend strata of a scaffold, again, because if scaffold being with a larger void, when you seed the cells, you might get more cells working all their way through. But again, we have protocols with modified seeding systems to kind of reduce that as well. So that is really the only thing that you might want to be looking after, but most cell type will be quite happy to grow on it. So as I mentioned before, we have compatibility with a huge amount of applications and routine application that people will be expecting to do with their 2D culture. Yes, we can do it in Alvetex. And here are just a few visual examples. So you can see for yourself, we can do a little bit of everything, and protein extractions look clean, RNA extraction look clean. You can even retrieve cells from 3D and do flow cytometry with it. Very good for microscopy because it's very stable. And this is a positive point of Alvetex compared to some other 3D system, which is it's very stable and it's lovely to handle, very easy to process, very easy to pick up with a pair of tweezers, to cut. It behaves itself very nicely in terms of material, so processing is actually quite easy and arguably a lot easier than some other techniques which are going to be a little bit more tricky and more difficult to handle during the processing itself. Here you've got an example showing you can do live cell imaging, so the depth is not an issue if you want to do imaging. The only thing is you will need to use confocal microscopy, but these days most people use confocal microscopy because it gives you such better imaging anyway, even for 2D. So really I can't think of a technique that really you would have to say, oh no, we definitely can't do it in Alvetex. You might have to use different protocols, and for most things we will have a protocol to help the customers, but again, the great majority of things is, yes, you will be able to do it, and we have a protocol on the website. So technical resources, this will be equally for yourselves, but also for the customers that you can direct them to. And it's about building the confidence in saying, yes, you can do this, 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 and here is how you can do it exactly in detail. We've got frequently asked question page, we have protocol pages, which is quite extensive. We have application notes, so these are more for research areas and detailed results, just to show the breadth and the quality of data that you can get with Alvetex, as opposed to protocols, which are really a detailed step-by-step, this is how you're doing it, and this is just an example picture. We have some videos which are quite good for an introduction to 3D cell culture, an introduction to Alvetex, things to direct the customers to. We have a few webinars for people who are interested in more in-depth applications. And also we have videos for demonstration of techniques, which are very simple techniques, things like how do you feed your culture, how do you actually take the disc out of the insert. So things that the customers can just go and have a quick look and convince themselves that yes, it's easy to do it, or they can just refer to it. In terms of publications, this is very important to us because our technology is very well supported, both by publication from our own lab originally, but more and more by customers, which is wonderful because these are people who are completely unrelated to us, use the product, found that it works, and have managed to publish it in a peer-reviewed journal. And this is really giving confidence to potential customers that you can get the quality of data that you would expect. And also, again, because there's more and more interest in the 3D field, it's actually a plus to your publication. You're probably more likely to get published if at least part of your work has been done in three-dimensional culture. And that is a nicely plus sell points for people who are not sure about whether they want to use 3D culture. And if all else fails and you can't find the information that you want on the website, you can always send me an email. So that's info@reinnervate.com. I will try to answer within 24 hours. And yes, and that is if you can't find the answer to what you want, either you can direct your customers to this email, which is me, or for yourself, whenever you need help, you have a question, however silly it might sound, just pop me a mail. And all of these and more, because our website is quite dense in information, is just accessible from our general website. So now we'll just go to the other side of this presentation, which is to look at more in-depth applications. This is more about the research areas in general that are quite popular with the customers that we are finding at the moment, but also things that you can make people aware of, and these are more the strengths of Alvetex, and this is a little bit more complex. And these are just a few examples. We have more application and there's more research areas, and again, if you just look on the website, you will find all the relevant information. So neural tissues, historically this is how our lab started. We're interested in stem cell differentiation, and here you can see this is stem cells in vivo, and you can see how they're forming those structures, which are like little round structures, and these are neural rosettes. And you can see here these are quite a good approximation of what you would find here in an animal. So that is showing you that this three-dimensional structure of Alvetex scaffold, in that case, is allowing you to replicate something you would have in an animal. This is very interesting to people who've got ethical concerns in their research as opposed to just practical concerns. One thing that I was mentioning before was the tissue slice maintenance on Alvetex strata. This is a section of spinal cord which has been put on strata, and that is going to be imaged through live cell imaging. Now that if this was done on a glass slide, you would see significant drift of the culture as you're going through your live cell imaging. And when you want to do live cells, you don't want something which is going to be moving around because it makes your life a lot harder to quantify. With Alvetex strata, you've got a tissue that is going to stay put where it is, and you don't have to worry about this drift. And this is an example of data that was done through collaboration with a university in Ireland, one of our customers. Another model that is very popular is the neurite outgrowth model. So this is a case where we have an aggregate of cells. Here you can see the center of the cells in blue, the nuclei. So it's a ball of cells that's been aggregated in vitro, not on the scaffold, but in vitro, and then which has been placed on the scaffold. And what we're looking at here is the generation of all those little filamentous structure, which are the neurites. So if you imagine your nerve cells with all the little neurite going, this is seen from the top of the scaffold, and you can see how all the neurites are just nicely radiating from the structure. This is from the side. You can see here the little blue ball of cells in the middle, which is staying on top, and the neurites in green are penetrating inside the scaffold as well as radiating on top surface. And this is the bottom, so the other side, and this gives you an idea that these neurites have traveled 200 microns through the whole of the substrate and are being now visible from the bottom. And this is a very good system to look at healing of neurites or differentiation of neurites, because you have a system where you can count the number of neurites on the bottom, put your drugs and see how that affects them. Is that going to stop from migrating? Is that going to increase their migration? Now, the idea of migration you could also apply to a cancer model, where you would put your cells on top and you would look at cancer invasion inside the substrate, and you could quantify that again. Now, this has got a good cross-selling potential within our mother groups, because obviously we are part of REPROCELL. Now, REPRONeuros, which are the induced pluripotent stem cells. So people who are interested, customers which are interested in neural tissue might be interested in buying the REPRONeuros. They also have Alzheimer's diseased cells. Again, very good models because the controls are available, so they might be interested in that. In some territories, the medium for growing the cell is available. And finally, we have a molecule which we developed here at Reinnervate, TC23, which is now available through StemGent. And this is a very good differentiation-inducing molecule. So if you're working with stem cells and you want to differentiate them into neural tissues, TC23 is a retinoic acid equivalent, which is a lot more stable and will give you a lot more robust differentiation. So, you might, if somebody's interested in neural tissues, they might be interested in Alvetex, but they probably also might be interested in those other products. So it's always good to try and help the sister companies. Another model which is very popular, and this is very popular mostly with industry people, which tends to have much more money involved into it, so we like our industry people, is the skin models. We have two skin models. One is epidermis only in Alvetex scaffold. These are the keratinocytes. So for people who are only interested in looking at epidermal cell response, this on the side, and it's an SEM, so scan electron microscopy, and you can see how the keratinocytes have really invaded through the whole of the scaffold here. And if you were going to look at the top, you're getting this picture, and this is flaking at the top of your skin. As you're thinking about how in vivo skin exfoliates and flakes, this is the equivalent that we are getting in Alvetex scaffold. So this is showing how good a model it is at replicating that skin surface. And that is obviously of interest for any kind of company that's got skin products. We've gone one step ahead and on a full thickness skin model. So this time the keratinocytes are on top of the scaffold, and here again you can see that layer, which is the stratum corneum, which is the bit that flakes and exfoliates in vivo, and that is replicated here in Alvetex. And underneath are fibroblasts, and the fibroblasts are producing collagens, and they are filling the scaffold. Fibroblasts are a lot more sparse, but this is perfectly normal. This is what we see in vivo as well. And here is a model where you've got your epidermis, but you also have your dermis. So it's a full thickness model. You can study interaction between dermis and epidermis, and we have a recent publication which is looking at essentially skin cancer. So they've replicated this model and then they've introduced skin melanoma cells here, so skin cancer cells, and looked at the invasion into the dermis. So this is more of a research, academical, health kind of application. So very good for industry, but also any kind of skin cancer and research. It's taken a very long time to develop, but it is now a very good model. Right. So the other one which can be of interest to industry, and this is more for ethical concerns now, which are being more and more important because customers want all their animal-free testing, is Alvetex for teratoma assay replacement. A teratoma assay is one of the benchmarks for knowing whether the cell line you've got can be called a stem cell. So you get your cells, you inject into an animal, and then you give it for so many weeks and you see what's happened to your cells, what kind of tissue they've differentiated into while they were inside the animal. And if you can prove with staining that they have differentiated into the 3 germ layers, which is the ectoderm, the endoderm, and the mesoderm, that is one of the tests to say, yes, it is a stem cell. And then you can market it as a stem cell and so on and so forth. Now, what we can do with Alvetex is do some things where we've got the aggregates, so those cells are grown in vitro on a low adherence petri dish or on a hanging drop system. And then you put them on Alvetex. Here you can see the Alvetex here, and you can see that's 200 micron. That gives you an idea of how big that structure is. And you will leave this aggregate here, and you leave it for quite a few more weeks to develop and to mature. And then you would do the same and do your staining. And you can see from these pictures the complexity of the tissue you're getting. This is a very nice picture of neural tissue. And here you can see how you've got different tissue type developing on here, and this is therefore all in vitro, and you don't have any need for an animal there. So it's a good alternative for either for labs which don't have access to animals, or for labs or industrial partners which are a bit more conscious of ethical issues and they want to do a first curl of their cell lines, the target cell, and say, well, we want to see which one are really interested in and reduce the animal uses. That is quite a good motivation to sell Alvetex to some of those people. So one thing that is quite useful to mention to the customer is the 3R initiatives. They should be familiar with that because for funding that is always a plus. It's reduce, refine, and replace, and it's all based on reducing animal use in research. That is one good reason for selling them Alvetex, is you can say you're doing something towards the 3R initiative, and that makes everybody else happy. Right, so obviously Alvetex, there's a lot of space in it. So one thing that you can do, which is quite exciting and which you cannot do in 2D, is looking at more advanced co-culture model. Co-culture being when you've got 2 different cell types being cultured in the same well. Because we have both insert formats and plate format, and because our discs can be removed quite easily from their formats, you've got a lot of different setups that the customers can play with. So here the cell, you've got one cell type in red, one cell type in blue. You could have both cells mixed and put into one disc, either on an insert or plate. You could have one in 3D on an insert, one in 3D on a plate, or you could have them just touching each other, and here at the limit you could look at what interaction the cells are doing, or maybe either whether one is going to invade into the other's layer. Here it would be more a setup where you would just look purely at the soluble factors, and you could look at the difference between the two to see what is mediated by soluble factors and what is going to be mediated by attachment factors. And you can have a look as well on 2D cells at the bottom and 3D cells at the top, or 2D cells on the top and 3D cells at the bottom. So this is a little bit more like the skin, like we were looking at, where you had cells growing in 3D and then cell growing on top. We do have within our protocols a number of protocols for coating. So we do have a protocol to make a gel at the barrier here at the top, so you can keep cell types separated if you need to. And here are just a few examples. So here are cancer cells on the top, fibroblast cells on the inside, and then you can follow how the cancer cells are going to be invading progressively within something which is not empty scaffold, but more a dermis model, as we had in the melanoma invasion model, which has just been published. Here is a neural model where you've got your neural aggregate on top, and here we've got glial cells which are growing inside the scaffold, and this is more to test more, for example, a glial scar kind of model. So we're getting a level higher up in complexity when it's not just what this cell type is doing, but how two cell type are interacting. And then, as we've seen in skin before, and this is a human airway, if you want tissue morphology, quite often in a tissue you would have several cell types with a very specific architecture, and having the scaffold in 3D allows you to replicate that specific architecture. So you can see cell behavior which is a lot closer to what you would have in an organ or in a tissue, as opposed to just cells mixed together in 2D. So here, if you remember, that is looking a little bit like the skin models, where you have fibroblasts growing inside the scaffold. In this model, they did a collagen gel in the middle, so that they didn't have to worry about the epithelial cells invading in, so that's an option. And here you can see the epithelial cells, which have nicely multilayered, and here there is a lot of mucus, which is what you would expect to be produced in a human airway. So the advantage of 3D here is architecture. You're managing to replicate something in vitro, which if you did a section of human tissue, you would get something which is very similar. And when you get the similar morphology, you can be a bit more confident that you're going to have similar function, and therefore a model which is a lot more relevant and where you can study a lot more complex processes, and where you can be confident that it is physiologically relevant. And that is the very big advantage of the 3D models. Now, obviously morphology isn't the only thing. We're also looking at functionality, and one thing which is very big for industry is drug testing and cytotoxicity and hepatotoxicity. So this is a test that would be necessary for every drug that is going on the market. You want to know whether your drug is going to be damaging the liver, or how it is going to affect detoxifying enzyme activity, because that might have an effect for polypharmacy, i.e. patients who take more than one drug. You want to know if they're going to interact with each other via the liver enzyme metabolism. And here we've got developed a 96 well plate format, as mentioned before. And you can see here that when we've put paracetamol, which is a well-known drug to affect the liver, Alvetex scaffold also shows an increased sensitivity compared to 2D. So here, by example, here is a drug where you could say, well, by having used three-dimensional culture, we know that this drug is going to affect the cell at a lower concentration than what we would have thought if it was 2D. And obviously this has got very, very important implication for human safety. Here is an example of detoxifying enzyme activity in 3 dimensions. So in Alvetex scaffold, you can see how there's greater detoxifying enzyme activity. And again, this is all about physiological relevance. And you know that your cells are going to work harder and more like what they would like in vivo. And therefore, it's a better predictive model for what you're expecting to see in an animal, what you're expecting to see in a human. And it's all helping in the drug pipeline. So this is of very, very good interest to industry. And to follow on from that, because we have a 96 well plate, we have worked with partners well known in the industry, Tecan and AstraZeneca, and this is working on automated systems. So automation is very big in industry, not so much in academia, but it's just to say, yes, it can be done. This is our plate, 96 well plate tend to be black, 384 as well, compared to the 2D, and it is perfectly compatible with the automation system. So again, this is about this routine use, something that the customer doesn't have to worry about whether our plates are going to fit their system. And here is a little bit of data just showing collaboration with AstraZeneca. This is very useful, again, to give confidence to customer, because everybody knows AstraZeneca, and if you can tell people, well, they've tried our plates, you know, if they've done it, that means that customer is probably can have the confidence to try it as well. So cross-selling potential, we are in the process of developing liver toxicity and cancer drug assay. This is between Biopta and Reinnervate. So this is in development, but this will be offered by Biopta as a service. So this would be for customers that they might not be very sure, or if they don't want to do it themselves, well, Biopta will be in a position to do it for them at a price. So that is something that we can offer and that we can cross-sell as well. And then finally, I don't think there's many, I mean, it'd be nice if we had more of our customers working on space exploration, but it's always a nice, cool one to kind of excite the customers a little bit. And this is a project that really was looking at bone development in zero-gravity condition and about bone loss. It was supposed to be relevant to people who have got very little activity and how bone loss and bone density is involved with that. So we were very lucky to collaborate with a lab that was involved with NASA. And you can see here we've got that little case with the cells and the scaffold being there. Medium was being pumped through syringes so it could go through the bioreactor. There was a little bit of fixative in there, so at the end of the experiment the samples could be fixed and then they could be analyzed later. So it's a very complicated and a very sophisticated piece of kit, and we're very privileged to be part really of it. So it was very exciting for us, and it's something that excites customer a lot. Right. So that was the applications that people tend to be asking us more about, that tend to be more popular, but it's not all. It's amazing everything you can do with Alvetex. So I've mentioned stem cells. You can either propagate them and maintain them on Alvetex, but also you can differentiate them, not just into neural tissue. We've done differentiation into bone and also into fat tissue. I've briefly mentioned cancer. You can look at cancer invasion through how far they go into the substrate, and this can be done through sections or through confocal microscopy. And you can look at toxicity as well, so drug testing for cancer. Primary cell culture, we can do that for liver, we can do that for neural tissues. Essentially, the advantage of doing primary cell culture is that instead of taking your cells from a 3D environment, which is in vivo, and putting them in 2D and therefore forcing them to that transition of flattening and changing their gene expression, you can put them straight into a 3D system. So they never really lose that three-dimensional environment and they don't have to go through that change of protein expression and gene expression. So they have been less modified compared to what you would have. So that is quite a good system to preserve the characteristics. We have a small number of customers which are interested in oxygen sensing, which is a very specific application, but it just shows what people can do with Alvetex even without our help. Epithelial models as well, especially with if you want an epithelium and then a submucosa underneath, so that's complex, a bit more complex system, but we can do that. And as I mentioned, we have a perfusion system, so some people can be interested. And of course, we can only think of so many things ourselves. And if you come across customers which have ideas, and they say, "Oh, well, I'd like to try for this, for that," we're always happy to advise as to whether we think that yes, indeed, it would be suitable, or if there's anything that needs to be modified first. And we do like to keep in touch with our customers who do something slightly different, because if that ever gets published, then we want to know about it so we can put it on the website, and that can drive other people in their field to have a bit more confidence in buying our product. So it's something we like to build good relationship with our customers. All right, this is everything. So that was a bit of a whistleblowing tour, but I hope you've enjoyed it, and if you've got any question, I'm here to answer them. Thank you very much.
Thank you very much, Fred. That was excellent. I just wanted to ask if anybody. Okay, so I actually have a couple of questions in the chat box. So the first one is, could you give us some examples when a customer should use Alvetex scaffold or Alvetex strata? So I know you went through it a little bit, but if
So, I mean, one very good example of the difference between scaffold and strata is, by example, if you want to retrieve your cells. When you retrieve your cells, you don't want them to be in a substrate which is going to get too much in the way of your detaching agent. So if you've got a very invasive cell line, I would say you want to retrieve them to do flow cytometry, go for scaffold, because the voids are big enough that the detaching agent will go in very nicely and can detach them fine, and you can retrieve them. If you were using strata, there might be too into because the substrate is smaller, it would be harder to get to those cells. So in that case, scaffold would be better. Conversely, if you've got an epithelial cell type, and you're pretty confident that they're not going to go into strata because they're not really invasive at all, they will probably multi-layer on strata. So you'll be able to access them a lot better. And a good example for that is liver cells, where they multi-layer and they will be on the top of strata and very accessible. But if they were in scaffold, because they're in a void, they're slightly less accessible. So if you've got an epithelial cell type you want to retrieve, grow on strata. If you've got an invasive cell type which you want to retrieve, grow on scaffold. Another example is if you want to maintain some stem cells or some primary cells in 3D before you implant them into an animal. Again, you want them to stay as they are as much as possible, so strata would be better because they're just going to sit there and be nicely maintained and be nicely fed because they're getting medium from underneath and from below, but they're not really going to be encouraged to move into the substrate. Well, if you had them in scaffold, they would just go and invade and just make the most of it, but you would have problem then getting them back out. If you wanted to test the invasiveness of different type of cancer cell line or different stages of a cancer cell from patients, strata would be better because again you would be able to make the difference between a slightly invasive cell line and a really invasive cell line. But then there are applications for which scaffold is better, so that if you want good three-dimensional growth, but you don't have a cell line which is highly invasive, you can just get into scaffold and it's going to go in and it will be nice and easy to process and to manipulate because it's just going to be in that scaffold. It's going to section nicely, it's going to stain nicely. So if you're just doing cells which are not really demanding, not really specifically demanding, scaffold is probably better. And then if you've got some specific applications which you need to think a little bit more about what is your cell structure or where is your cell invasiveness, then strata is what you want. And for core culture as well, scaffold is better because you can establish your dermal or your submucosal equivalent within the scaffold. The cells are going to go in, it's not a problem, you don't have to think about needing them to be very invasive, and then you can have your other epithelial cells on top. So it was a long answer to a short question. Sorry about that.
No, thank you. Thanks, Brett. I think another question that Vanessa's posted is Alvetex compatible with organoid culture, for example, as published by the Noblish Lab recently by Johns Hopkins University?
Yeah. I'm not familiar with that specific paper, but based on our experience on tissue slices and on maintenance of aggregates and of maintenance of embryonic bodies and so on, I would say that's another one where strata is good. Because again, there's no substrate to get in the way. Therefore, if you've got substructures which you want, like you want specific tubules, you want specific cell organization within your population, they don't have anything to worry about. You don't have any substrate in the way. They're just going to sit there and do it by themselves. Like everything else, you might be limited in terms of growth, so you can't grow them things indefinitely, because at some point you would start having problems of feeding. But again, that can be good for other models, like cancer models, where it's known that you get necrosis in the middle of a culture because they don't have access to the blood on the outside. So that can be something she's doing. But again, that's one where strata might be better than scaffold. But it should be compatible, yes.
Right.
Yeah, so in this case, the Knoblich lab, which is on the rise with the stem cells, and that you culture them, you grow them in a 3D organate culture. So this was the example with the mini brains. Yeah, just to get a more relevant differentiation of the cells and interaction of the cells.
Yeah, but I mean, as I said, I haven't seen that paper, so I don't know how complex they got it. But definitely, like in the teratoma assays where the aggregate go on top, we do get cells differentiating and expressing neural markers. Obviously, the more complex you want your structure, the more difficult it is to get the morphology right. Scaffold is very good when you've got a layered morphology, like skin, like any kind of submucosa. We haven't tried retina yet, but where anything is in straightforward layers, scaffold's really good. When you start talking about more complicated structure, like we have customers asking about gills structures and tubule structures and spheroids, even biocanaliculi for the liver, then the strat is probably better because that's where you get the maximum amount of cell-cell contact, and they can really interact and do their stuff without any interference.
Okay.
Great.
Any more questions?
No, not at this point, I think. But if anybody has any questions, they can always be sent to info@reinnervate.com.
Absolutely. Yeah, exactly. And I'll get back with them. So we're always happy to have questions.
Thank you. Okay, thank you very much. So I'll close the meeting now, and please get in touch with any queries you may have. Okay, thank you.
Thank you. Bye.